Dear students, today we will be discussing about microsporangium. Microsporangium. Before going into the subject, we need to understand some basic things for easy understanding. What is microsporangium? Where it is located? What is the structure of microsporangium? What is the function of microsporangium? All those things. Then only we can easily understand the topic. Okay, now we will see the location of microsporangium first, where it is located. In the last video we discussed about the structure of typical angiosperm flower. In that it is thalamus, and thalamus in the first whorl we saw the first whorl, that is sepal or calyx. Second whorl is corolla. Okay, third whorl androecium. Fourth whorl gynoecium. Here the calyx and corolla, these two are not involved in sexual reproduction. That's why we call them as non-essential parts, non-essential organs. Okay, but the third and fourth whorls are very important. They involved in sexual reproduction, that is androecium and gynoecium. That's why these two whorls are very important in flower. Androecium is a male reproductive organ, gynoecium is female reproductive organ. Now we are starting with androecium related thing, means male reproductive organs are related things. The microsporangium also, it is related to androecium. Okay, but where exactly the microsporangium is located, that also we will see now. Before that, what is androecium? Androecium is a third whorl of the flower. Third whorl of the flower. It consists male reproductive structures called, it consists many subunits. Each subunit is called stamen. Okay, each subunit is called stamen. Okay, now we will see about stamen structure. Stamen consists two parts. One is the basal thread-like or tube-like structure, filament. Apical bulged or lobed structure called anther. It consists two parts. One is anther, second one is filament. What is the function of filament? Filament keep the anther position at different heights. Based on the species, it will increase in length so that it plays in different heights. Okay. Now, anther is fruitful part. Actually, in the future, it will produce pollen grains. These filament also supply food materials and water to this maturing anther. Okay, it is attached somewhere to the anther called fixing. Okay, based on this attachment, anthers basically classified into 3 or 4 types. Now we will see what type of anthers and how they are attached to the anther. Okay, if the filament, sometimes the filament attach at the base of anther. At the base of anther, it is very basic type. Okay, here the attachment is at base. These anthers are called base-fixed anthers. Okay, sometimes the attachment will be different part, like it is anther. The attachment on dorsal surface in the middle or somewhere here. This attachment or this type of anthers called dorsi-fixed anthers. Okay. Now one more type also we can see, that is the attachment will be dorsal side, but here the attachment throughout the dorsal surface, means the filament will attach entire surface, dorsal surface of the anther. That's why the filament and anther attached entire throughout the dorsal surface. These condition are these type of anthers called adnate anthers. Adnate anthers. And some anthers or some filaments attach another type. It is also a type of dorsifixed anther only, but the attachment is very with very a tip of filament, means it is needle tip like structure, but the anther size is very big. That's why the anther will loosely rotate or dangle in the air, like it rotate clockwise and anti-clockwise. That's why the pollen will spread easily. Usually the wind pollinated flowers follow or having this type of anthers. Usually, for example, in grasses we can see this type of versatile anthers. The type name is called versatile anthers, versatile fixation or versatile anthers. This is the classification of anthers, basic type of anthers based on the attachment of or fixation of this filament and anther. Okay, now we coming to the anther types. Even anthers also classified into 2 or 3 types that we will see based on the lobation. Lobe means this is a lobation. Based on the number of lobes, anthers are classified into different types. Usually the anthers are two-lobed in angiosperms. Two-lobed means like this. In the cross section we can easily understand. Whenever we take a section of it, we will see the structure anatomy like this. Means it consists 2 lobes. This is one lobe, this is another lobe, second lobe. Okay, like this the lobes will be 2 in bilobed anthers. But in case of anther, the lobe classification, we need to understand one thing. Whenever we call lobe, one lobe equals to 2 locules. 2 locules. If one lobe means it consists 2 locules. Here we call bilobed, 2-lobed anther. That's why it is equals to 4 lobes. It consists 4 lobes, means locule or locules. Here in the corners we will see locules. But lobes are 2, locules are 4. How? Because each lobe consists 2. It is unique for all types. Okay. Now this is the universal type, means it is common type throughout all angiosperms. But occasionally we can see one-lobed anthers. One-lobed anther means it consists only one lobe. In the anatomy, we can easily understand whenever we observe the anatomy. In the anatomy, we can see only 2 locules, means only one lobe. That's why we will see 2 locules. This is occasional type in angiosperms. Usually in Malvaceae family, we will observe this. This is common type. Even Solanaceae, in all types of most of the families consist these 2-lobed anthers or 4-loculed anthers. But the 2-loculed or 1-lobed anthers are uncommon, but occasionally it is also found in some families like Malvaceae. But rarely we can see 4-lobed anthers also. 4-lobed anthers means the anthers will be like this. Whenever we observe the anatomy, you can understand very well about these 4-lobed anthers. Like this, the lobes are 4. Each consist 2 locules. Each consist 2 locules. So that it will be calling as 4-lobed or 8-loculed anthers. It is very rare. Usually you can see this type of anthers in Bixaceae. What is Bixaceae? Bixa, it is a one genus which is the color is produced from the Bixa plant. You may search in Google, you can find so many images about Bixa. In that we will see 4-lobed or 8-loculed anthers. This is a rare phenomenon in Bixaceae. Now one more type also, it is extremely rare. 1-loculed anthers means not even one lobe, only it consists only one locule. It is very rare phenomenon. You can see in one plant called Arcithobium minutissimum. It is a plant name. In this plant we can see only 1-loculed anthers, means anther consists only 1 locules, not even one lobe. And next, to understand Microsporangium, we need to understand the anatomy of anther. That's why now we are discussing about anatomy of an anther. So that just we are discussing about two-lobed anther here because it is a common type of anther in angiosperms. That's why we are discussing about two-lobed anther. Two-lobed means it consists 4 microsporangia in the anatomy. Means like this: 2 lobes, 4 microsporangiate anther. Okay, this is the anther which is common among angiosperms. That's why now we will see the anatomy of two-lobed anthers. Okay, now I will draw one diagram. The anatomy clearly show 2 lobes and 4 locules or microsporangia. Okay, this is outline. In the 4 corners you can see a circular structures called microsporangia. These 4 are very similar to each other. That's why we will do with this so that we can extrapolate this thing to remaining part also. Okay, now we will see the anatomy of this part. Okay, here in the anatomy, the first you can see one layer of cells as usual, as like in all plant parts. Here also the outermost layer, the one cell layer is known as an epidermis. It is an epidermis. It is a outermost layer in the anther. Okay, after epidermis again we will see one more single layer like this, but cells are somewhat radially elongated like this. How these layers are evolved or developed, that we will see next to this, because after doing this anatomy we can see the development of these cell layers also. This is second layer which is having a prominent nucleus also. Okay, this is second layer. This layer is called as endothelium. Endothelium, it is a second layer. After this a cell layer having horizontally elongated cells. Horizontally elongated cells. Usually these type of cells having 2 rows, means they are arranged in 2 rows. These 2 rows together called as middle layers. Middle layers. These are the third and fourth oral in this anatomy. Okay, middle layers. Usually you can see 2 layers of these cells. After that a cell layer consists a very big cells with dense cytoplasm and with clear nucleus, prominent nucleus, and some granule like structures. We will discuss what are these structures little later. Okay, this is the innermost layer here. That layer is called tapatum. It is a tapatum. Okay, it is the innermost layer. After this we can see a dense cell mass more or less uniform in size and shape. These cells also having clear nucleus and also rich in nutrient materials. Okay, these these cells are called sporogenous cells. Sporogenous cells. This circular structure is known as sporangium. Sporangium. Okay, where the pollen grains will be developed just in the future. Okay, from sporogenous tissue or sporogenous cells, later we will get pollen grains. Okay, and usually one more thing, the anatomy of anther can divide it into two major parts. They are including epidermis, endothelium, middle layers and tapatum. These four layers. These four layers together called as anther wall. Very important, anther wall. Okay. The inner part is other one, that is sporangium. These four layers together called as anther walls. The innermost circular part is known as sporangium, where the pollen grains will develop. Okay, it is an anther wall which give nutrition and protection to this sporangium. Okay, now we will see other parts also. The same thing, the same anatomy will be here also and here also and here also. Same, the four corners consist the same anatomy as it is, but in the center you can see a dense cell mass consists very compactly arranged cells called connective or connective tissue, where the filament actually attach to the anther. It is a place where the filament is attached to this anther. It is nothing but it is useful to transportation of water and minerals. Through this, the anther get water and minerals. Okay, that's why it is connective. It is present here. Remaining all tissue is loosely arranged parenchyma tissue only, but here it is densely arranged a transportation tissue. Okay, like this. But here one more important point is, here will be connective tissue, here common parenchymatic tissues, but the anther wall only restricted to these portions only. Anther wall develop only in these parts only. Remaining part is just having one type of tissue called a simple parenchyma. But from here to here, the anther wall will be developed. Means the cell differentiation like epidermis, endothelium, middle layers and tapatum. These cell variation we can see only in these zones only. That also you need to remember. Again we will discuss about anther wall. It is very important topic, and also what is the function of each layer, that also we will discuss. This is anatomy of anther. Okay, now the same layers we will be discussed here again as under anther wall and their layers. Okay, we already discussed the same thing here, but now I'll just maximize here. The image is maximized here. Okay, in the anther wall we have four layers. We have four layers. That is, the first layer is epidermis. Next to the epidermis, there will be one more layer of cells. These cells are usually somewhat radially elongated. Radially means in this direction. It is also a single layer called endothelium. Endothelium. This is second layer in anther wall. After that, a horizontally elongated cell layers. Usually there will be two layers in typical anthers. Okay, two cell layers. These two cell layers together called as middle layers. Middle layers. After that, the innermost layer in the anther wall, that is a big cell layer. It is also a single cell layer in the anther wall, having very prominent nucleus, rich in nutrients and dense in cytoplasm is very dense. This cell layer is called as tapatum. Tapatum. This is all about anther wall. Anther wall consists 4 layers. The outermost layer is epidermis. What is the function of epidermis? That also we will see now. Epidermis is the outermost layer which is having only one layer of cells. As usual, as typical epidermis, here these epidermis also giving protection to the internal tissues. Internal tissues. That is the only function of epidermis here. Okay. The second layer is endothelium. It is also a single layered cell layer. Okay, having only one cell layer thickness. It is placed next to the epidermis. Here the walls consist fibrous thickenings. Fibrous thickenings. Usually the fibrous thickenings more in the maturity stage. These fibrous thickenings, since they are present in the cells, they having the cell layer having hygroscopic nature. Hygroscopic nature. Since they are having these fibrous thickenings in the cell wall, this layer having hygroscopic nature, which is useful to the anthesis. Anthesis means breaking of anther. After maturity, due to this hygroscopic nature, it will useful to the dehiscence of anther. Okay, that is the function of this endothelium. After that, middle layers. They are horizontal cell layers, usually 2 in typical anther wall. These middle layers sometimes may reserve some food materials. That food materials transfer to the tapatum. Whenever the scarcity of food materials occur in the tapatum, the middle layers can transfer that food material to the tapatum, usually during the pollen development. Okay, this is about middle layers. Usually we can see 2 layers of middle layers. Next, innermost layer. Next layer is tapatum. Tapatum, it is a innermost layer in anther wall. It is very important layer also, because it provide nutrition to the developing pollen grains. Nutrition to the developing pollen grains. That's why it is very important layer, which is having very big cells. The size of tapatum cells is very big, having dense cytoplasm, rich in nutrients. That's why they behave like, or they act like nutrition layer to the pollen grains. Okay, this is a function of tapatum layer. Now, based on the studies, we have 2 types of tapatum cells. One type is called secretory tapatum, or glandular tapetum. This is one type. Second one is plasmodial type or amoeboid type. Okay, means the tapetal cells are classified into two types. In some plants we can see secretory or glandular type of tapetum. In some plants we can see plasmodial type or amoeboid type of tapetal cells. It is also known as nuclear type or invasive type of tapetum also. It is secretory or glandular tapetum. Here the only difference here is whenever they are providing nutrition to the developing pollen grains, they just, in case of secretory cells, both are having nutrients, okay, nutrient materials which are reserved for pollen grains. But here the difference is, in case of secretory or glandular tapetum type, they just secrete or release or ooze the nutrient material into sporangium or into sporogenous tissue. These secreted nutrition is taken by pollen grain and they grow luxuriously. That is in case of secretory tapetum type. But in case of plasmodial or amoeboid type, here there is no secretions. Entire cell wall will be degraded here, release all cell contents into sporogenous tissues, so that the cytoplasm will invasive into the sporogenous tissue. At the time we can also observe lobes or pseudopodia like structures. That's why it is also known as amoeboid or plasmodial or pseudopodial type tapetum or invasive, because they are invading into the sporangium. That's why invasive type of tapetum or nuclear type of tapetum. Why? Because nuclear type means whenever these cell walls broke up, what happened? They all nucleus will come together. We can see a xenocytic nature. Xenocytic means multicellular nature, multinuclear nature. That's why it is also known as nuclear type of tapetum. Okay. After releasing these cell contents, utilized by these pollen grains. Okay. Like this, the nutrition will be gained. But here cell walls remain as it is. They just release or secrete the cell nutrient materials into the sporogenous tissue. That secreted materials will be utilized by these cells. After releasing all nutrients, the tapetal cells are become shrink or become very small, because all materials released out. That's why they become small. But in case of plasmodial type or amoeboid type, here the cell walls are literally dissolved, so that entire cell content will be flooded into sporogenous tissues. There is a difference between these two cell types. Here, one more important point about the exclusive character or characteristic feature of secretory tissue is the presence of ubished bodies. These are the nothing but spherical or globular structures present in secretory type of tapetum cells. It is only exclusively these ubished bodies exclusively found in these tapetal type only. But in this type, the ubished bodies are absent. Only in the secretory or glandular type of tapetum cells only having ubished bodies. These are nothing but a spherical structure, very minute in structure. The size is about few microns in the radius. Okay. The ubished bodies, the function of ubished bodies not yet clearly known, but two peoples. One is Panchanan Maheshwari. He is father of Indian embryology. he stated that it may useful to the development of exine of pollen grain. Exine of pollen grain. Exine, what is exine means? If it is pollen grain, the outer layer is called as exine. Inner layer is called intine. He opined that, or he believed that they may function as to useful to the production of exine layer of pollen grain. That is the opinion of Maheshwari. Later, the other scientist called Heslop Harrison, and Dickinson. In 1969, they said that this is not these Ubish bodies are not done any work related to pollen grain. They just supply sporopollenin, or they just transport sporopollenin into these sporangiums. That is only function. This is the statement given by Heslop and Harrison. And also Heslop and Harrison, he called these Ubish bodies as orbicules. Heslop Harrison, as per his opinion, he named them as orbicules. It is name given by Heslop Harrison. Again, Heslop and Dixon, they proposed one more statement or theory that the Ubish bodies are structures useful to transport sporopollenin, which is very important component in the development of pollen grain. This sporopollenin is transported by these Ubish bodies. Okay, that is all about Ubish bodies. Here we need to remember three scientists' names: Dickinson, Heslop Harrison, and our Panchanan Maheshwari. Okay, they talked about Ubish bodies. The tapetal types also very important. Two types of tapetal cells. One is secretory or granular. Another one is plasmodial type or amoeboid type or invasive type or nuclear type, whatever it may be. Okay. Contrastingly, it is called as cellular type also, because cells remain forever, just releasing the nutrients. That's why it is otherwise called cellular type. Okay, this is all about the innermost layer, that is tapetum. Now we have four layers: epidermis, endodermis, middle layers, and tapetum. Okay, this is all about anther wall. Now we discussed about the anatomy of a typical anther, that is two-lobed anther, and also the clear anatomy of anther wall. After that, we will see the sporogenous tissue and the division of sporogenous tissue and the formation of microspores or pollen grains. Okay. Now we will see the structure of sporangium. Actually, it is a globular structure. Now it is tapetum layer. These cells are sporogenous cells. After maturity, these sporogenous cells, they divide mitotically, they will give MMC, means microspore mother cells. This is first step. The sporogenous tissue undergone so many mitotic divisions, they can give MMCs. MMCs means microspore mother cells. These microspore mother cells, again, they meiotically, or they undergone meiotic division and gives rise 4 cells from each. Means one MMC, means one microspore mother cell can undergone meiotic division so that it will give 4 cells. From 1 cells we will get 4 cells. Okay, these 4 cells after meiosis they are not divide individually, though they are completely the wall formation and completely they are divided, they just remain together as a groups, as a group. That group of 4 cells is known as tetrad. Okay, it is common phenomenon in all angiosperms, that is after meiosis it will be like that only, means fuse it together for some time. This structure is called tetrad. The formation of tetrad, it may vary among plant to plant. Usually we can see 4 types of tetrads in angiosperms. One is all are tetrads only, but based on the arrangement of these cells or pollen grains, we can name them in different way. This type of, whenever they are arranged, means one side we can able to see 3 pollen grains only. Whenever the tetrads are arranged like this, the shape is called tetrahedral. Tetrahedral. It is very common tetrad formation in angiosperms. Very common. 90% of plants showing this type of tetrad formation. But sometimes the cell may divide into like this, so that the 4 cells will be appear from one side. This tetrad is called isobilateral. Isobilateral. This is one more type of tetrad formation. Sometimes it form another tetrad type, that is decussate. Decussate means 2 cells they placed horizontally, 2 cells they placed vertically. That's why 2 cells placed side by side. These 2 cells placed horizontally, means this side 1 cell, that side 1 cell. This is called decussate type of tetrad. Decussate type of tetrad. In some plants, the tetrad may appear like a T-shaped manner, because 2 cells are in anticlinal manner, they divide in a anticlinal manner. Some 2 cells they divide or they placed horizontal manner. These cells are horizontal or periclinally they divided and gives 2 cells, and 2 cells are anticlinally divided and 2 cells are placed here like this. That's why it is called as T-shaped tetrads. T-shaped tetrads. Sometimes they can also form eye-shaped or linear tetrads also. Means all cells or all daughter cells can placed in one line also. That's why we can see linear tetrads. Linear tetrads. These are the common type of tetrads we can see in the angiosperm, but the most commonest tetrad type is tetrahedral type. Okay, this is all about tetrad formation. After tetrad formation, due to the secretion of some enzymes from tapetal and middle layers, and due to the some enzymes, these cells, okay, these cells will be divided into individuals. After division, each tetrad will give 4 microspores. Okay, these microspores are nothing but pollen grains, but in the immature stage. They are in immature stage. These microspores again getting the tapetal secretions or materials from tapetal layers, they fully develop so that they transform into full-fledged pollen grain. pollen grain, after formation of pollen grain, at the time the entire wall layers will be almost in the end stage. Okay, after full maturity of pollen grains, they burst somewhere. Somewhere means in the stomium position, the place called stomium. In the stomium place, the anther will burst and the pollen grains will be released. These pollen grains is otherwise known as male gametophyte. Male gametophyte, because it produces male gametes. That's why it is called male gametophyte. Microsporogenesis. We already discussed that one microspore mother cell, that is MMC, will undergo meiotic division, so that we are getting 4 microspores or pollen grains, nothing but. Okay, we are getting 4 microspores from one meiotic division. But here, how the cytokinesis is taking place? Based on that, we may observe 2 types of cytokinesis during this meiotic division. There, the first type is successive type of cytokinesis. Second one is simultaneous type of cytokinesis. Here it is very simple. Whenever the first meiotic, for example, this is MMC. Okay, generally the meiosis occur in 2 stages, meiotic 1 and meiotic 2 phase. Okay, now based on the time of wall formation, these 2 types were divided. Here in successive type of in successive type of cytokinesis, the pollen mother cell undergone meiosis 1 followed by wall formation. Means, for example, it undergone meiosis 1, immediately the wall formation may take place. Okay, immediately after first division, it will take the wall formation will take place. After this, in the second division also, even in the second division also, immediately again the second phase, wall also formed immediately after division. Okay, that's why we get 4 cells. But in case of simultaneous type, the first meiotic division of MMC, that is pollen grain mother cell or microspore mother cell, whenever it divide meiotically, the first division not followed by wall formation, means not followed by wall formation, so that only karyokinesis only taking place, means only nucleus will divide into 2, the cytoplasm remain only 1. Okay, after that again one more division, that is second phase of meiotic division also taking place, so that we will get 4 nuclei within one cell. Finally we will see the wall formation. Okay, so that we will get 4 cells. Here also 4 cells, here also 4 cells only, but the difference is when the wall is forming, either it is immediately followed by the first meiotic division or after second division. That is a basic criteria. Based on that, we have 2 types of cytokinesis or 2 types of divisions here. Okay, first in the successive type, after immediately first division, we will get wall formation in between 2 daughter cells. After second division also, immediately we will get wall formation in between daughter cells. But here the wall formation is taken at last. First they will divide, only nucleus will be divided. There is no wall formation or septa formation in between these 2 daughter cells. After second division, at the last they will again 4 walls or 2 walls, so that 4 cells will be produced. Here also 4 cells, here also 4 cells, both are produced tetrads only, but the formation of wall, the timing of wall formation is criteria here. Okay, this is all about anther anatomy, anther wall structure, layers in anther wall, microsporangium structure, microsporogenesis and tetrad formation. In the next class, we will be discussing about the development stages of Anther Wall and Microsporangium. Thank you.